Hello everyone. So today I'm going to explain on the topic of development of nervous system. Before I begin, let me ask you a quick question. Have you ever wondered how our brain and spinal cord form before we even born? It's amazing to think that the entire nervous system starts as just a thin layer of cell in an embryo. Today I'll take you through the fascinating journey of the development of nervous system from a simple embryonic structure to complex system that control everything we do. So let's see. Introduction to nervous system. What is nervous system development? The nervous system developed from a germ layer called the ectoderm. The sequence is first fertilization happens. First fertilization happens and gastrulation happens, and then the formation of ectoderm happens. Then neural plate formation happens. The neural tube is formed. Think of it's like a building a house. Before building a house, we first need a blueprint. Similarly, before forming a brain, the spinal cord, the embryo first creates a basic blueprint called the neural plate. The development begins in third week of embryonic life and continues throughout the fetal development. The final result is nervous system capable of controlling sensation, movement, thinking, memory, and emotions. Now we'll see the formation of neural plate. Well, the formation of neural plate. Now let's see how neural plate forms. A structure called the notocode acts like a teacher giving instruction. It sends signaling molecules to the ectoderm and say it's time to become nervous tissues. As a result, the ectoderm thickens and forms the neural plate. A quick question for all of you. First, which germ layer forms the nervous system? If you've corrected the ectoderm, it's right. Now we'll see neurulation. It's the formation of neural tube. This is one of the most important stages. This process is called neurulation. Imagine folding a sheet of paper into a tube. Similarly, the neural plate develops a groove called the neural groove. The side arise from the neural folds. The folds come together. They fuse and create the neural tube. Easy trick to remember is the plate to groove and groove to folds and folds to tube. Well, repeat it with me. Plate to groove, groove to folds, and folds to tube. The neural tube later forms the brain and spinal cord. Clinical connection is if the tube fails to close properly, the anterior neuropore failure, it is known as anencephaly, and posterior neuropore failure, it is known as spina bifida. Now, the derivatives of neural tube. Once the neural tube is formed, the different parts develop into different structure. Think of the neural tube as a train with two ends. The front end, cranial end, becomes the brain. The back end, caudal ends, become the spinal cord. The cranial end develops into midbrain, forebrain, and hindbrain. This later form important adult structures such as cerebrum, thalamus, pons, cerebellum, and medulla. Interesting one. The hollow space inside the neural tube remains throughout the life and forms brain's ventricular system and central canal of the spinal cord, allowing circulation of cerebrospinal fluid. Development of brain vesicles. We can see the brain vesicles. Now the brain becomes more specialized. Initially, there are three primary brain vesicles: prosencephalon, mesencephalon, and rhomencephalon, respectively known as forebrain, midbrain, and hindbrain. Later, they become five secondary vesicles. Easy memory tip: we can say it is forebrain divides into two, telencephalons and diencephalons, and midbrain stay once, mesencephalons. Hindbrain divides into two, again metencephalon, myelencephalon. How many secondary brain vesicles are formed? If you have guessed five, it is right. By the fifth week of development, all five are established. Then we'll go to neural crest cell. The neural crest cells are the neural crest cells are the cells which separate from the neural tube and migrate throughout the body. I like to think them as the construction workers carrying materials to different sites, you can say. These cells form sensory ganglia, autonomic ganglia, Schwann cells, adrenal medulla, melanocytes, craniofacial structures. Yeah, this is like that. So we'll forward move to congenital abnormalities of nervous system. What happens when development does not occur normally? as we have studied earlierly, spina bifida. Incomplete posterior neuropore closure of the occurs, which leads to vertebral arc defects and spinal cord exposure. Anencephaly, it occurs due to failure of anterior neuropore closure, and it results into absence of major brain structures incompatible with life. Then hydrocephalus occurs when cerebrospinal fluid accumulates because of obstruction, causing enlarged ventricles. The important point from this is the best preventive measure is we should take folic acid supplementation, which reduce neural tube defects. Now we could conclude nervous system developed from ectoderm, neural plate forms during third week, neurulation process produce neural tube forms CNS, neural crest cells form much of PNS, brain develop many primary and secondary vesicles, neural tube